There is a group of people living inside the United States right now. No smartphones, no electrical power, no combustion engines, traveling by horse and carriage along roads built for a world they deliberately refuse to join. They have maintained this way of life for 3 centuries without interruption, without compromise, and without apology. And buried inside their cells is a genetic signature that has no business being there. Not according to history, not according to geography, not according to any framework scientists brought with them when they first began to look. When researchers set out to map the DNA of this community, they carried a clear and reasonable expectation. What they would find, they believed, was exactly what 300 years of strict internal marriage and deliberate isolation should produce. A genome compressed by repetition, narrowed by ancestry, defined by its sameness. A biological portrait of a people who had turned inward and stayed there. What the data actually showed changed the assumptions quietly but completely. Take any state highway through Lancaster County, Pennsylvania, on a weekday morning, and you will experience something that doesn't fit cleanly into the present tense. Horse-drawn black buggies rolling slowly along the shoulder of roads, used by 18-wheelers and pickup trucks. Men wearing broad hats and suspenders. Women in modest dresses and white prayer coverings. Fields worked with draft animals rather than machines. Gardens maintained by hand rather than irrigation systems. The rhythm of the day still organized around sunrise and sunset rather than screens and schedules. This is Amish country, and it looks almost exactly as it did a century ago. In many ways, it looks as it did 2 centuries ago. The Amish arrived in North America in the early decades of the 18th century, fleeing a wave of religious persecution that had targeted Anabaptist communities across the German-speaking territories of Europe for generations. They were pacifists, separatists, people who believed that the Church of Christ should exist apart from the institutions of worldly power, and who had paid for that belief in exile, imprisonment, and death. Pennsylvania, with its promise of religious tolerance under William Penn, offered them something they had never had in Europe, the freedom to be left alone. They took that freedom seriously. They settled in clusters, built plain meeting houses, planted farms, and began the work of building a community designed to last. Their spoken language was a dialect of German, today called Pennsylvania Dutch, that preserved features of 17th-century speech that have long since vanished from the European continent. Their surnames, Stoltzfus, Yoder, Byler, Lapp, King, Esch, Fischer, trace back to a small and specific cluster of villages in Switzerland and Alsace. Their theology, their dress, their agricultural practices, their relationship to technology, all of it has been transmitted across generations with a fidelity that no other community in the Western world has managed to match. Every family in the Amish world, by both historical record and community tradition, descends from the same original group of founding settlers. Estimates vary, but most scholars agree the founding population numbered somewhere between 200 and 500 individuals. This is the story that everyone agrees on. It is not inaccurate. It is simply incomplete in a way that turns out to matter enormously. The practice geneticists call endogamy, marriage exclusively within a defined group, is one of the most powerful forces in human population genetics. It is not unusual. Many communities around the world practice some form of it, shaped by religion, ethnicity, caste, or geography. But the Amish have practiced it with a consistency and duration that is extraordinary by any measure. They do not marry outside the community. They do not bring in converts. New members occasionally join, but in numbers so small that their genetic contribution to the overall population is negligible. What this means, in biological terms, is that the founding gene pool has been circulating almost entirely within itself for roughly 12 to 15 generations. The mathematical consequence of this is well understood. In population genetics, when a small group founds a new community, and that community remains reproductively closed, the genetic diversity of the founding group gets compressed over time. Certain variants become more common, others are lost entirely. Rare recessive mutations, which would be diluted to near invisibility in a large open population, begin to appear with much higher frequency because the same limited set of carrier ancestors appears in nearly every family tree, often on both sides. This is what geneticists call a founder effect. And it is precisely what makes the Amish medically fascinating. Researchers have identified a remarkable cluster of rare genetic disorders that appear at elevated rates among Amish populations: metabolic conditions, cardiac abnormalities, neurological syndromes that are vanishingly rare in the general population, but show up with unsettling regularity in communities where the same founding variants have been cycling for centuries. Studying these disorders is where formal Amish genetics research began, and it is where the unexpected discovery first surfaced. The anomaly did not announce itself dramatically. It appeared as a quiet irregularity in data that researchers at Johns Hopkins University were processing in the early years of the 21st century. The team was doing what seemed like routine work, mapping the genomic architecture of Amish populations to understand how certain recessive mutations concentrated and spread across family networks. They were not asking questions about ancestry or admixture. They were asking clinical questions about disease. But when the population structure data was processed, something appeared in the background that had no place in a closed Swiss-German colonial community. The Amish genome, according to the data, carried the signatures of outside genetic contributions, what geneticists call admixture, from populations that should not have been present in a sealed religious colony founded by Central European Anabaptist refugees. The researchers noted it, they discussed it, they ran the analysis again. And then, as often happens when data contradicts the expected narrative, they moved on. The assumption was artifact, a processing anomaly, a contaminated reference panel, a statistical ghost. Not the kind of thing you build a paper around when you are trying to publish findings about metabolic disease in pediatric patients. But the signal did not go away. It appeared in separate studies conducted at different institutions. It appeared in different geographic clusters of Amish communities, in Pennsylvania, in Ohio, in Indiana, in the smaller settlements scattered through the Midwest. It appeared in individuals with no known recent shared ancestry, which meant it could not be attributed to a single family line carrying an unusual variant. It ran through the population like a thread woven into fabric from very early on. Consistent, recurring, distributed. 3 possible explanations gradually took shape, and each one pointed toward a different version of history. The first explanation was the most conservative, and it started from a simple observation. Colonial America was not a well-organized place. The early 18th century, when the Amish were establishing their first permanent settlements in Pennsylvania, was a period of extraordinary social fluidity on the American frontier. Records were incomplete. Communities overlapped in ways that later, more orderly generations would find difficult to imagine. English colonists, German-speaking settlers, French fur traders, free black individuals in the mid-Atlantic colonies, Scotch-Irish migrants moving through the backcountry. All of these populations were in motion simultaneously, in a landscape with no fixed administrative order and no reliable system for tracking who went where or lived with whom. The theory, in its simplest form, was this. In the first 1 or 2 generations of Amish settlement in America, before the community's internal boundaries had fully hardened, a small number of outsiders entered the genetic lineage. Not through any dramatic event, not through anything that would have been recorded or remembered, just through the ordinary human reality of living in proximity to other people, of relationships formed before the rules were fully established, of exceptions made and forgotten. A few such events, scattered across the founding decades, could have introduced outside genetic material into several family lines simultaneously. Once that material was inside an endogamous population, it would not dilute as it would in an open community. It would stay. It would circulate. By the time the community had fully sealed itself, which happened gradually, not all at once, those threads would already be woven in. This was a satisfying theory. It was tidy. It explained the anomaly without requiring anything dramatic, and it had the advantage of being consistent with what we know about the general messiness of early colonial settlement. The problem was the data. The admixture signal was not behaving the way leakage at the edges behaves. It was not scattered unevenly across family lines, concentrated in some clusters and absent in others, the way you would expect if the source was a handful of chance colonial encounters. It was too coherent. It was distributed with a consistency that suggested the contribution had entered the population early, deeply, and across multiple founding lineages, not crept in at the margins after the community was already established. Something else had to be considered. The second explanation reached backward across the Atlantic. Before the Amish ever sailed for Pennsylvania, their spiritual and biological ancestors, the Swiss Anabaptist communities of the 16th and 17th centuries, were living in one of the most historically turbulent corridors in all of Europe. The Rhine Valley, Alsace, the Palatinate. These territories were not backwaters. They were the crossroads of a continent that had spent 3 centuries tearing itself apart through plague, religious war, and dynastic conflict. The 30 Years' War alone, which ended in 1648, displaced millions of people across Central Europe, destroying communities and scattering populations across national and ethnic boundaries in ways that were never systematically recorded. The people who survived that war and ended up in Swiss Anabaptist communities in the 1650s and 1660s came from a landscape that had been churned and reshuffled to a degree difficult to comprehend today. The theory here was straightforward. Perhaps the founding population that eventually became the Amish was never as genetically uniform as either its own traditions or the historical record suggested. Perhaps the ancestors who gathered in those Swiss valleys in the years before the Pennsylvania migration already carried admixture from populations further east, further south, further removed from the Swiss-German core. Populations absorbed into European communities over centuries of migration and conflict that left biological traces even when they left no written ones. If this was the case, the anomalous genetic signal in the modern Amish genome would not be American at all. It would be European in origin, ancient by the time the first ships left for Philadelphia, embedded in the founding population before a single Amish family had ever seen the Atlantic. This was a genuinely compelling possibility, and it was also the hardest to test. It required going back to the source, comparing modern Amish genomes not just against Western European reference populations, as earlier studies had done, but against a broader and more precisely calibrated set that included Eastern European, Balkan, and Near Eastern lineages. It required genetic data from European Mennonite communities that had never emigrated, from Swiss and Alsatian populations descended from the same geographic areas as the Amish founders, from the full range of populations that might have contributed to the Central European gene pool during the chaotic centuries before 1700. That kind of analysis takes time and resources and collaborative infrastructure that did not exist in the early years of this research. And while researchers were working toward it, the third theory emerged. The third theory was the one that refused to be dismissed. In 2021, a research team affiliated with the University of Pennsylvania completed an analysis that had not been attempted at scale before. Rather than comparing Amish genetic data only against the Western European populations that historical accounts identified as the community's source, they ran the comparison against a much wider reference panel, one that explicitly included Eastern European populations, Central Asian lineages, and, critically, reference genomes derived from pre-Columbian indigenous American ancestry. The paper was published in a specialized journal. It did not receive mainstream press coverage. It was written, as genetics papers must be, in language hedged carefully with probabilistic qualifications and calls for further study. The authors were cautious, appropriately so, about the implications of what they were reporting. But what they were reporting was this: a consistent subset of genetic markers appearing across multiple Amish family clusters, in individuals living in different states, with no established close common ancestry in recent generations, showed statistically clear affinity with populations of indigenous American descent. Not the kind of background affinity that connects all human beings across deep evolutionary time. Not the faint signal that reflects shared ancestry from tens of thousands of years ago, before human populations diverged into the lineages we recognize today. Something more recent, more specific, more localized in time. The statistical modeling that accompanied the paper estimated the timing of this admixture event, whatever it actually was, as falling somewhere between the 15th and 18th centuries. That range is wide enough to encompass several different scenarios, but it is narrow enough to rule out ancient history. Whatever produced this signal happened within a few centuries of the present. within the historical era, within a period for which records, however incomplete, exist, which meant it could have happened in Europe, or it could have happened here. Before the Amish settlers arrived in Lancaster County and the surrounding territories in the early 1700s, that land was occupied. The Susquehannock people had lived along the Susquehanna River Valley for centuries before European contact. By the time Anabaptist colonists were establishing farms in the Pennsylvania interior, the Susquehannock had been severely diminished by decades of epidemic disease and conflict. But other Algonquian-speaking and Iroquoian-speaking communities were still present, still navigating the rapidly changing social landscape of the colonial mid-Atlantic. The Amish relationship with indigenous neighbors in this early period was genuinely different from that of most other European colonial groups, and understanding why that difference existed matters here. The theological core of Anabaptist belief, non-resistance, pacifism, rejection of violence in any form, meant that the early Amish and Mennonite settlers approached their indigenous neighbors with a posture unlike the confrontational and often murderous relationship that characterized English colonial expansion. This did not mean the Amish were innocent of the broader structures of colonial dispossession. They benefited from land arrangements that displaced indigenous peoples regardless of how peacefully individual interactions proceeded. But at the level of daily contact, trade, labor, shared use of resources, neighboring, there is documentary evidence of sustained interaction between Anabaptist German-speaking settlers and indigenous communities in the Pennsylvania interior during the 18th century. What has never been systematically studied is whether any of that contact produced biological connections. A single union, a child, an individual absorbed into the community under circumstances that were never formally recorded, because the community's own record-keeping was never designed to capture that kind of event. In a community defined by strict endogamy, that would not need to happen many times to produce a genetic signal that persists across centuries. It would need to happen once, early enough, in a family line broad enough that the contribution circulated before the population fully closed around itself. But there is another path this signal could have traveled, and it runs in exactly the opposite direction. In the 16th and 17th centuries, the period before Amish immigration began, the period when the Swiss Anabaptist communities that would become the Amish were taking shape, a small number of indigenous people from the Americas were present in Europe. Their presence there was the result of a range of circumstances, most of them coercive. Some were brought to European courts as curiosities or diplomatic symbols by the colonial powers returning from the New World. Some were transported as part of the brutal human trafficking that accompanied European expansion. A small number arrived through more ambiguous arrangements, accompanying traders, missionaries, or explorers in roles that the historical record describes only partially. What happened to these individuals once they reached Europe is largely undocumented. Many died quickly, unable to survive in an environment where they had no immunity to common European diseases. Some lived for years. Some, the historical record suggests in fragmentary ways, integrated into European communities, marrying, having children, disappearing into the social fabric of cities and towns in ways that their descendants would have had no way of knowing about and no reason to report. The Swiss and German-speaking territories where Anabaptist communities were concentrated in the 1600s were not isolated from this phenomenon. These were connected, commercial, religiously active societies with access to the networks through which indigenous people moved across Europe in this period. It is not impossible, it is not even particularly improbable, that one or more individuals of indigenous American ancestry were absorbed into a Swiss Anabaptist community in the decades before the Pennsylvania migration. If that happened, the genetics of closed communities would have done something remarkable. In an open population, a single genetic contribution from one unusual ancestor dilutes rapidly across generations. Within 4 or 5 generations, it may be statistically undetectable, but in an endogamous community, the mathematics work differently. The gene pool does not expand outward to absorb and dilute unusual variants. It turns back on itself, cycling the same limited set of ancestral contributions through generation after generation. An unusual genetic signature introduced early into such a community does not fade. It circulates, It may even grow more concentrated over time, as the same limited founder pool is drawn from repeatedly. This is the same mechanism that concentrates rare recessive mutations and produces the elevated disease rates that first attracted researchers to Amish genetics in the 1990s. It works on all genetic material equally, whether that material codes for a metabolic enzyme or simply marks a line of ancestry that connects backward to a person whose name no one in the community knows. An event that occurred in 1640 or 1660, a single individual, a single family, absorbed into a community that was already developing its practices of separation and mutual enclosure, would not have been erased by time. It would have been preserved by the very same mechanisms the community used to preserve everything else it valued. It would still be there, in 2024, in the genome of an Amish farmer in Holmes County, Ohio, who has never left his county, never used a smartphone, and has no way of knowing what his cells contain. Think for a moment about what it would feel like to be a researcher sitting with this data for the first time. You have spent years working in clinical genetics. Your relationship with the Amish genome is fundamentally practical. You are trying to understand how rare diseases spread through closed populations, trying to help families understand their children's diagnoses, trying to map the inheritance patterns of conditions that mainstream medical research rarely bothers to study because they don't appear in large enough numbers outside these specific communities to attract funding. It is important work. It is careful work. It requires building trust with a community that has good historical reasons to be cautious about outside institutions. And then, on an afternoon that begins as ordinary as any other, you are running a population structure analysis as part of a larger data set. The software is doing what it always does, sorting genetic variation into components, assigning ancestry proportions, generating the visualization that shows you how much of a given genome traces back to which reference population. And something appears that you cannot account for. You check the sample label, you verify the reference panel, you rerun the analysis with different parameters. You ask a colleague to look at it without telling them what you already see, to confirm you are not projecting something onto ambiguous data. The signal does not go away. It is, by every measure available to you, real, consistent, statistically significant, and it has no place in the story you have been working from. The history books do not contain it. The community's own genealogical records do not contain it. The founding narrative that has organized your understanding of this population since you began studying them does not contain it. But there it is, written in a language older than any archive in any library in the world, waiting patiently to be read. The picture that is emerging from this ongoing research, and it is still emerging, still being contested, still being refined by researchers working from different methodological approaches and different theoretical starting points, is not a simple one. The Amish are not a simple thing to begin with. They are, in the most genuine and profound sense, the living heirs of a specific, documented, traceable European religious tradition. Their theology, their language, their community structure, their practices of mutual aid and collective discipline, all of this connects backward in an unbroken chain to the Swiss Anabaptist communities of the 1520s and 1530s. The people who established those communities faced violence and death for their beliefs. The people who carried those beliefs to Pennsylvania did so because they had no safe place left in Europe. That history is real, and it is theirs. And yet the genome that carries this history also carries something else. Something that arrived by a route the historical record has not preserved. Something that was absorbed into the community, whether in the Rhine Valley in the 1650s or in Lancaster County in the 1720s, whether through a single encounter or several scattered ones, and then held in place by the same forces of endogamy and communal closure that the Amish brought to bear on every other aspect of their inheritance. Geneticists use the word palimpsest to describe a document that has been written over multiple times, where older layers of text show through beneath the newer writing, where the accumulated history of the parchment is visible to anyone who looks carefully enough. The Amish genome is a palimpsest. It is a document written by many hands, across many centuries, in several languages, only one of which was ever acknowledged by the people who carried it forward. The significance of this finding is not about hierarchy. It is important to say this clearly. because genetics has historically been misused in ways that turned ancestry into a ranking system, as though some lineages were inherently more valuable or more legitimate than others. That framework is not science. It is ideology dressed in scientific language, and it has caused incalculable harm. Indigenous ancestry, European ancestry, African ancestry, these are not categories of greater or lesser worth. They are simply different pathways through human history. All of them ancient, all of them connected to communities and individuals who lived full lives and built meaningful worlds and deserve to be remembered with respect, rather than reduced to data points in someone else's story. What the Amish genetic discovery illuminates is not the relative value of any ancestry. What it illuminates is the nature of communities themselves, what communities actually are, as opposed to what they believe themselves to be. The Amish have built the most deliberately bounded human community in the modern Western world. Their theology of separation, Gelassenheit, they call it in their German dialect, a word that means yielding, submission, the release of individual will into collective identity, is not merely a social preference. It is a cosmological commitment. The boundary between the community and the world outside it is not a practical convenience. It is a sacred line, maintained at enormous cost, across extraordinary pressures, for 3 centuries without interruption. And even so, even within this community, perhaps the most closed, most intentional, most disciplined human project of self-definition that the modern world has produced, the world got in. Not through technology, which the Amish have guarded against with exceptional thoroughness, not through ideology, which they have resisted with a consistency that has outlasted every modern movement that once seemed sure to absorb them. Not through any of the channels they knew to watch. It got in through the simplest, oldest, most irresistible force in human history, the tendency of human beings to make contact with other human beings, somewhere, in some moment that no document records and no memory preserves. 2 people whose languages were mutually unintelligible, whose worlds were separated by an ocean or by the frontier of a new continent, whose communities had no framework for acknowledging what they were to each other. Those 2 people produced a connection that the body carried forward when everything else was lost. And the child, or the grandchild, or the great-grandchild of that connection was welcomed into a community that would carry it in complete silence for 300 years, until a laboratory instrument finally gave it language. There is a question that lives underneath all of this, and it is not really a question about genetics. It is older than genetics by thousands of years. If the Amish, who have tried harder than any comparable community in the modern world to remain exactly what they are, to preserve without dilution the identity that their founders carried from Europe, if even they contain within themselves the traces of encounters their history has no room for, then what does that tell us about the rest of us? About the certainty we feel when we describe where we come from? About the confidence with which we draw the lines of our identities and say, inside this line, my people. Outside it, other. Every family tree to the people living inside it feels like a closed and legible document. Every community that has ever existed has understood itself as bounded, coherent, and knowable. Every nation, every ethnicity, every religious community, every clan has told itself a story about its own origins that emphasizes continuity, purity, and unbroken transmission from a specific, identifiable past. The genome, patiently and without editorial opinion, suggests otherwise. It always has. The genome is not a record of isolation. It is a record of movement, of the relentless, irreducible human tendency to cross every boundary that other humans have drawn, across rivers and mountain ranges, across linguistic barriers, across religious prohibitions, across the formal and informal rules that communities have always maintained to define themselves against the world outside them. The genome records all of it, impartially, in a language that does not recognize the categories we have built to make sense of ourselves. The Amish did not know what was written in their DNA, and in not knowing, they preserved it with greater fidelity than any archive could have managed. It traveled forward through 3 centuries of deliberate stillness, held in place by the same discipline of communal enclosure that the Amish believed would protect them from the world outside. In a sense, it did protect them, but not in the way they meant. It protected the memory of a meeting that history forgot. It kept the record of a human encounter that crossed every boundary the world had drawn, and that proved, as all such records do, that the boundaries were never as solid as the people maintaining them believed. The machines in the laboratory did not create this story. They only made it audible. It was always there, traveling forward in the cells of people who had no idea they were carrying it. Through generations of plain dress and plain speech and plain devotion, waiting for the moment when someone would finally think to ask a different question.